Welcome students, hope you are well and doing great. This video is on the chromosome and gene linkage, and it contains the instructive and educational content that is both shareable and retained. So let's start. First of all, we will see the introduction of the chromosome. The word chromosome is derived from the Greek word chroma means color and soma means body. It is defined as a microscopic thread-like part of the cell that carries the genetic information. That means it is the fundamental part of the cell that contains the all genetic information. It is always present in the form of pairs, and they pass from parents to their offspring, that the one generation to their next generation. The one of the basic property of the chromosome is its compactness. It is always present in the compact form that had to organize it during the cell division. When cell division occurred, its compactness helped it to organize itself. For example, there are 23 pairs of chromosomes in the human body in the compact form. Here you can see the structure of the chromosomes. It is a thread-like structure that is coiled around and they form an organized structure. You can see the arms of the chromosome, and it is a well-defined structure of the chromosome. Let me discuss about the feature of the eukaryotic chromosome. It is become visible during the cell division. It varies in size, shape and number, and it is composed of the three basic units: DNA, RNA and protein, and it is always present in a well-defined nucleus. Here is some difference in the eukaryotic chromosome and prokaryotic chromosome. As we know that the prokaryotic cell don't have a well-defined nucleus, so their chromosome is not present in the nucleus. While in case of the eukaryotic chromosome, as we know that there is a well-defined nucleus in the eukaryotic cell, and their chromosome present inside the nucleus. As we know that the number of the chromosome varies from animal to plants and species to species. Here is the example. Human have the 46 number of the chromosomes in the 23 pairs, and rat, onion, pea, and cat, they have, you can see the different number of chromosomes in different species. All species have a different number of chromosomes. The structure of the chromosomes, it is mainly made up of the basic components that are the centromere, telomere, chromatids, chromomere, and the matrix. In the structure, you can see the telomere, centromere, their arms, chromatid, and DNA. Let me discuss in detail. Centromere it is the localized region of the chromosome at which the spindle fiber attach. Telomere it is the tip or the end of the chromosomes, and the chromatid, one half of the chromosome, is known as chromatid. The one of the two longitudinal subunits of the chromosomes is known as a chromatid. The chromomere means some species have a bead-like structure on the chromosome, which is known as a chromomere, and the matrix it is the fluid part in which chromosome is present. Here you can see the centromere, chromatid, and tip is the telomere, and there is a specialized protein which is known as a kinetochore. The chemical composition of the chromosome, as we discussed earlier, that it is made up of the basic three units, which is the DNA, RNA, specialized, and they have the two types in case of chromosome, which is the histone protein and non-histone proteins. In the structure, you can see the chromosomes and the proteins, ball-shaped proteins. DNA is coiled around. The DNA have the genes. Let me discuss about the types of chromosomes. On the base of position of the centromere. As we know that the centromere is the localized region at which the spindle fiber will attach. And on the position of the centromere, there are the various type of the chromosomes. The telocentric, it is the type of the chromosome in which the centromere is present at the tip of the chromosome. No arms will be formed because centromere is present at the tip. you can see in the structure. And while in case of the acrocentric, the centromere is slightly away from the tip से थोड़ा सा नीचे होता है, जिसकी वजह से आर्म्स बन जाती हैं, very short arm and the long arm. In the structure you can see easily. Next are the metacentric and submetacentric types. In case of metacentric type, the centromere is present exactly in the center of the chromosome and equal arms are formed. In the structure you can see the centromere is present in the center, while in case of the submetacentric, the centromere is slightly away from the center. Center से थोड़ा सा ऊपर और थोड़ा सा नीचे होगा, जिसकी वजह से unequal arms बनेंगी, P arm and Q arm. In the diagram you can see the difference in the telo, acro, submeta and metacentric type of the chromosome. Here you can also see the telomere is the tip of the chromosome and the centromere is a localized region, and here spindle fiber will be attached during the cell division, and the types are also mentioned here. There are other type of the chromosomes which are the major types. It is the autosome chromosomes and the sex chromosomes. Autosome chromosomes are those which are not involved in the sex other than sex characteristics. They are the autosome chromosomes. There are 22 pair of the autosome chromosomes in the human, while the sex chromosomes are those that are involved in the sex determination. They will be determined whether the organism will be the male or the female, and there is only one pair of the sex chromosome in the human being. X and Y chromosomes are the sex chromosomes. In the picture you can see. And some other types are the diploid chromosomes and the haploid chromosome. Diploid chromosomes are those which are always present in the twin form, and they are the result of the mitosis. As we know that the mitosis, one cell is divided into the two cells, and there is always present in the 2n diploid form of the chromosomes. All the autosome chromosomes are the diploid in nature, while the haploid chromosomes are those which are in the 1n form, and they result from the meiosis. In the meiosis, we know that the one cell is divided into 4 daughter cells and the number of chromosomes divided, and only 1n form are come into existence. All the chromosomes are the haploid chromosome because they are divided by the meiosis, and from one cell to 4 new daughter cells are formed, and they have only single chromosomes. Homologous chromosome and non-homologous chromosomes are also the type of the chromosomes. In the form of the homologous chromosomes, they are the member of the same pair, while in case of non-homologous chromosomes, they are not member of the same pair. In the picture you can see it is the pair of the chromosomes, and they are the homologous chromosome because they are the member of the same pair. The function of the chromosome, as we described earlier, it is the genetic information. It carries all the genetic information of the cell, so it contains the genetic material DNA. It helps in the development of the individual. It is the vehicle of the heredity because it contains the DNA. It protects the DNA from the damage. All the process which are in the living organism is controlled by the chromosome, so it is in charge of all the process. The cell division, replication, formation of daughter cell is also controlled by the chromosome. It is a packaging material that binds the DNA and the protein. Our next topic is the gene linkage. It is the tendency of the genes to remain together in their original combination during the inheritance. जब पेरेंट्स से ऑफस्प्रिंग को जेनेटिक मटेरियल ट्रांसफ़र हो रहा होता है, तो कुछ जीन्स ऐसे होते हैं जो एक साथ ही रहते हैं, वो सेपरेट नहीं होते, तो वैसे के वैसे ही अगले नस्ल में या अगली जनरेशन में ट्रांसमिट हो जाते हैं, और उनकी वजह से कोई रिकम्बिनेशन, कोई वेरिएशंस नहीं आती, बिकॉज वो एज इट इज़ ही अगली जनरेशन में ट्रांसफ़र होते हैं. नेक्स्ट, द करेक्टरिस्टिक ऑफ द जेनेटिक लिंकेज. इट कंट्रोल द ट्रेड्स. फॉर एग्जांपल, अगर पेरेंट्स में ट्रेड ये है कि उनकी आइज़ कलर्ड हैं, ब्लू कलर या कोई और भी कलर, ग्रे कलर, तो अगर वो उनके जीन्स वैसे के वैसे ही उनके ऑफस्प्रिंग में ट्रांसमिट हुए हैं, तो आगे उनके ऑफस्प्रिंग के भी बिल्कुल ऐसे ही आई कलर होगा। They control two or more traits. Traits may be recessive, dominant, homozygous, heterozygous. The gene linkage group is the group of the alleles that are close together and tend to transmit together. वो एक साथ ही ट्रांसमिट होते हैं अगले जनरेशन में, और वो उनका जो एक्सप्रेशन है वो बिल्कुल वैसा ही रहेगा। फॉर एग्जाम्पल जीन फॉर द रेड हेयर. There are the two type of the gene linkage. One is a complete linkage and the second one is the incomplete linkage. In case of the complete linkage, alleles at the same loci never get separated. वो अलग नहीं होते, एक साथ ही नेक्स्ट जनरेशन में ट्रांसमिट हो जाते हैं। जब वो एक साथ ही ट्रांसमिट होंगे तो कोई रिकम्बिनेशन नहीं होगा, कोई वेरिएशंस नहीं आएंगी। While in case of the incomplete linkage, alleles at the same loci get separated. वो अलग हो जाएंगे एक दूसरे से और रिकम्बिनेशन और वेरिएशन का चांस बढ़ जाता है इस केस में। Here you can see the crossing over between the non-sister chromatids. Homologous chromosomes हैं, they are the chromosome of the same member, और लेकिन इनके जो chromatids हैं वो नॉन सिस्टर, इनके सेगमेंट्स अलग हो जाते हैं। DNA का एक्सचेंज होता है बिटवीन द नॉन सिस्टर क्रोमाटिड्स, और जिस वजह से रिकम्बिनेशन और वेरिएशंस अगली जनरेशन में आ जाती हैं। Here is the significance of the gene linkage. द इफ़ेक्ट ऑफ सिलेक्शन, एक ट्रेट जो है वो पेरेंट्स से वैसी की वैसी ही ऑफस्प्रिंग में चली जाएगी, उनकी नेक्स्ट जनरेशन में जाएगी, तो ये एक सिलेक्शन का इफ़ेक्ट कॉज करता है। दे मेंटेन फॉर द जनरेशन्स। फॉर एग्जांपल अगर एक जनरेशन में कोई ट्रेट जो है वो मुसलसल चलती आ रही है, पेरेंट्स में कोई करैक्टर मुसलसल चलता आ रहा है, तो वो वैसे के वैसे ही लिंक्ड जीन की वजह से अगली जनरेशन में ट्रांसफर हो जाता है, तो वो पूरी जनरेशन के लिए मेंटेन रहता है, उनकी आने वाली नस्लों के लिए वो करैक्टर मेंटेन रहेगा। इट प्रिवेंट्स अ रिकम्बिनेशन, जब जीन जो हैं वो एक साथ ट्रांसमिट हो जाएंगे वैसे के वैसे ही, तो रिकम्बिनेशन का चांस बहुत कम हो जाता है। अगर लिंक्ड ग्रुप है, अगर इनकम्प्लीट लिंकेज पे बना रहा है तो तब वेरिएशन के चांसेस आ जाते हैं। Here you can see the genetic recombination. नॉन सिस्टर क्रोमाटिड्स हैं, उनमें क्रॉसिंग ओवर होगी, उनका जेनेटिक मटेरियल एक्सचेंज होगा, और वो एक्सचेंज जो है अगले उनकी जनरेशन को मिलेगा, तो उनमें कुछ चेंजेस अकर हो जाएंगी और रिकम्बिनेशन अकर होगा, और उनमें फिर वेरिएशंस का चांस आता है।